Greetings to everyone. So today my topic is replication of DNA. Objectives of this topic: importance of replication, types of replication, characteristics of replication, stages of replication and components required, steps involved in replication, differences between prokaryotic and eukaryotic replication, and finally inhibitors of replication. So we can see one by one. Now, replication of DNA. Why is there a need for replication of DNA? What is the need for replication? Replication is needed for new cell formation. And when does it occur? During S phase of cell cycle. I'll deal with cell cycle later. Now, this is the central dogma of life. Now, DNA should undergo replication for new cell formation. DNA contains the entire genes which has to be transcribed. So from DNA, formation of messenger RNA through the process called transcription, and from the messenger RNA, the corresponding proteins are translated, proteins are synthesized. That process is called as translation. So DNA contains the genes. Those genes are transcribed. So the messages from the gene are contained in the RNA, and from the messages in the RNA, proteins are synthesized. This is the central dogma of life. Now, what is replication? It is the process of synthesis of daughter DNA from parental DNA, and the process is semi-conservative. The site is in prokaryotes, cytoplasm, eukaryotes. It occurs in nucleus. So what are the various types of replication? So the process of replication may be semi-conservative, conservative, or dispersive. So semi-conservative, what do you mean by semi-conservative? Here you have the parental DNA strands, double-stranded helix. So one of the parental DNA and one of the newly synthesized daughter DNA, they form a duplex. This is semi-conservative because one of the parental strand is conserved in the newly formed daughter DNA. So it is semi-conservative. Conservative means after replication, after the end of replication, the two strands of parental DNA, they reunite, forming the parental DNA, and newly synthesized DNA strands, they join together, forming the daughter DNA. So here the parental DNA is conserved. So this is conservative DNA replication. Dispersive means both the newly formed DNA and the parental DNA, they form fragments, and those fragments are condensed together to form a DNA double helix. So these are the three forms. But the type of replication commonly occurring in humans is semi-conservative. What are the characteristics of replication? As I already told you, it is semi-conservative, semi-discontinuous. This you will understand when we see the actual process of replication. So semi-discontinuous, that is, during replication, one of the strands will be synthesized continuously and one of the other strand will be synthesized discontinuously. So that is semi-discontinuous and bidirectional. Replication occurs in both the directions, and high fidelity, it is highly accurate because of the proofreading property of DNA polymerases. So these are the four characteristics of replication: semi-conservative, semi-discontinuous, bidirectional, and high fidelity. Now you can see the semi-conservative nature of the DNA replication. This is the newly synthesized DNA and this is the parental DNA. So one of the strands is conserved here. So it is semi-conservative. This is bidirectional. So when the replication starts from this side, it will go on synthesize in this direction and also in this direction. So this is bidirectional. So overview of replication. So first target the site of initiation of replication. Then at the site of initiation, the double-stranded DNA will start unwinding. Then formation of the replication complex, reforming double-stranded DNA of parent and daughter strands, then reformation of the chromatin structure including nucleosomes, that is packaging of DNA. Organization of DNA finally occurs. Now we have three stages of replication: initiation, elongation, and termination. What are the various steps involved in replication? So first, the origins of replication has to be identified. So first, identification of origins of replication, that is, how does this replication starts? So first, origin of replication is identified, and at the site of origin of replication, unwinding starts. So single strand DNA is formed because only single strand DNA can act as a template. Template means having this as the parental strand. Complementary base pairs to this strand will be synthesized by the DNA polymerase. So this single strand DNA will be acting as a template on which complementary base pairs are bonded. Then formation of the replication fork, initiation of DNA synthesis, and then elongation. So next is the elongation step after formation of all these. Then formation of replication bubbles with ligation of newly synthesized DNA segments. Finally, reconstitution of the chromatin structure. We can see one by one. So first is initiation, origin of replication. The origin of replication at the consensus sequence. What do you mean by consensus sequence? Short sequence composed almost exclusively of AT base pairs. So these AT base pairs facilitate melting. Why does this facilitate melting? Because it contains only two hydrogen bonds. AT base pairs has two hydrogen bonds, whereas GC contains three base pairs. So AT-rich region will be easily melting. So this consensus sequence contains AT-rich base pairs which facilitate melting. In prokaryotic organisms, you have single site of origin of replication, whereas in eukaryotes, multiple sites along the DNA helix for rapid replication. Here you can see this is circular chromosome in E. coli. So in prokaryotes, circular chromosomes. So AT-rich region here, this is AT-rich region here. This is the origin of replication. So to this origin of replication binding proteins will come and bind to this site. So once these proteins bound together, origin replication complex forms, and then AT-rich region starts denaturing. So after denaturation, there will be melting of the double strand, formation of single strands. To prevent re-annealing of these strands, there will be binding of single strand binding proteins. You can see here. So denaturation occurs here, melting of the AT-rich region, so single strand binding proteins. This prevents re-annealing of this double strand because both are complementary to each other. So they can easily form the hydrogen bonds. So once origin of replication is identified, these proteins will come and bind to this site and the melting starts. So it separates into single strand DNA, and then to prevent re-annealing, single strand binding proteins bind to the single strand DNA template. So the origin of replication varies with different species. For example, in Bacteriophage lambda, that site is called as Ori lambda, origin of replication lambda. Binding protein is lambda encoded O protein. So this is 150 to 250 base pairs with multimers of the DNA binding protein. In E. coli, origin of replication is Ori C and the binding protein is DNA A. In yeast, what happens? Origin replication element is usually autonomously replicating sequences, which are 11 base per sequence. Then origin recognition complex, which is origin replication element plus DNA binding proteins. Then DNA unwinding element is origin replication element with adjacent AT-rich sequence. So because only this AT-rich sequence unwinds. So origin replication element is nothing but autonomously replicating sequences. The sequences are the origin replication element. Unwinding element is that plus adjacent AT base plus AT-rich sequence. Then recognition complex is along with the DNA binding proteins. So in circular bacterial chromosomes, what happens? They contain 6 into 10 power 6 base pairs of DNA. So single origin of replication. Replication completes in 30 minutes. Rate is 3 into 10 power 5 base pairs per minute. What happens in mammalian genome? It contains more number of base pairs, 3 into 10 power 9 base pairs. But this is completed in 9 hours. If single origin of replication as in circular bacterial chromosome, this will be completed only after 150 hours, but it's completed in 9 hours. How this happens? This is overcome by the bidirectional replication and multiple origin of replication sites. So these two are very important for completion in 9 hours. So you can see here single origin of replication in the bacterial chromosomes. Circular chromosome, single origin of replication and local unwinding happens here. Whereas in case of linear chromosome, as in mammalian genome, multiple origin of replication sites. So multiple replication sites. So this opens up here, and so here what happens? It opens here. The replication will be in both the direction. So in this direction and in this direction, at the same time in this site, origin of replication site, again in this direction, in this direction. So the replication goes on like this, so it's completed in 9 hours. The unwinding of double-stranded DNA to single-stranded DNA template, that is melting, separation and unwinding. Why this is essential means the polymerases, which is the enzyme used in synthesizing new DNA nucleotides, adding nucleotides to the DNA. This polymerase enzyme can use only single-stranded DNA as a template. It cannot use a double-stranded DNA as a template. So this melting and separation and unwinding is essential. And annealing is prevented by, as I said previously, by single-stranded binding proteins. Now, when the DNA, double-stranded DNA, when the tightly coiled DNA starts unwinding, there will be supercoil formation beyond the site of unwinding. So those are relieved by the enzymes topoisomerases. Proteins, what are the various proteins involved in replication? You have helicases, which is the enzyme for unwinding of the DNA helix. So this is ATP dependent. Then single strand binding proteins, you already know, topoisomerase relieving the torsional strain to release the supercoils. Then primase is the enzyme which helps in synthesis of RNA primers. This I will come to later. Then DNA polymerases, synthesis of new strand. DNA ligase, which seals the nick, that is formation of the phosphodiester bond between the nascent chain and Okazaki fragments in the lagging strand. So here in this picture you can see, this is the replication fork. So DNA helix enzyme. This is ATP dependent. So DNA helix, it is like a zip. So you are opening the zip here. So this is like a zip. It unwinds by opening the zip, helicase enzyme. So this is the direction of movement of the replication fork. And these are the single strand binding proteins. So now, when the helicase enzyme opens the double-stranded, it's tightly coiled. The DNA double helix is tightly coiled. When the helicase enzyme opens the zip, starts unwinding of the double helix, what will happen? there will be supercoil formation beyond the site of the unwinding. This you can see for yourself, taking a rope, tightly coiling it and tightly fixing at one end. You can start unwinding. You can see the supercoil formation here. Even in the telephone wire you can see. So this is the site of unwinding and these are the supercoil formation. You can see here how this happens. So starting unwinding, there will be supercoil formation. Now this is relieved by topoisomerase enzymes. So supercoils, as the double helix separates from one side during replication, supercoils are formed on the other side. That is released by topoisomerase. And in topoisomerase, you have two types, type 1 and type 2. Type 1 DNA topoisomerases reversibly cut a single strand of the double helix to overcome the problem of supercoiling. Both nuclease and ligase activities are present in the type 1 topoisomerase and they do not require ATP. You can see here only one strand cutting occurs here by type 1 topoisomerase. This will release the tension here and they start releasing the supercoil. And then finally, after releasing this supercoil, the nick is sealed by the ligase enzyme. Ligase activity present in the topoisomerase. Now, type 2 topoisomerases are also the bacterial type 2 topoisomerases or otherwise known as DNA gyrase. They make transient breaks in both the strands. You can see here, it's wound here. This is the left half, it folds over the right half. What happens? Type 2 topoisomerase cuts both the DNA strands. It cuts it, then it wound over the cut. So now you can see one coil is released and the released coil now it is the front half passes through the break which is resealed as in the previous. So this type 2 topoisomerase causes double strand break and type 1 topoisomerase single strand break. They relieve both the supercoils. Now what is the clinical importance of topoisomerases? Anti-cancer agents like etoposide, they target human topoisomerase 2 and antibiotics or antimicrobial organisms, quinolones. For example, ciprofloxacin, they target bacterial DNA gyrase, type 2 topoisomerase, so prevent replication in the bacteria. So this is the clinical importance. So till now, we have seen about the initiation of replication. So we have seen about the unwinding enzyme helicase, how the supercoils are released by topoisomerases, and how the origin of replication is targeted. Next is elongation. Now for elongation, the requirements are DNA template, which we have single-stranded DNA template now after initiation, and then the precursors to build the nucleotides in the new strand. We need all the bases, all the triphosphonucleotides, adenine triphosphatide, then guanine, thymine, and cytosine. Next, we need one important oligonucleotide, which is a primer with free 3' hydroxyl group. Why do we need the primer here for elongation? Because DNA polymerase enzyme cannot start synthesizing the newly strand by complementary base pairing of the existing DNA template. It needs one 3' hydroxyl group free end to add on the incoming nucleotides. So for that we need a primer. So now this is the reaction of the DNA polymerase, that is it needs a primer with oligonucleotide. So n number of nucleotides here with the 3' free hydroxyl group here. Now the incoming nucleotide is having alpha phosphate group. So this is added, incoming nucleotide triphosphate. This is having alpha phosphate. So this condenses with this 3' hydroxyl group, forming one additional nucleotide to the already existing oligonucleotide. So This is synthesized with another 3' 3 hydroxyl group. And from this triphosphate, pyrophosphate is released. This is the reaction happens in DNA polymerase reaction. So this is the template strand. This is the template. You can see the template strand. And now this is the primer having 3' hydroxyl free end. Now these nucleotide triphosphates are added to this. So complementary base pair to this template strand is added here by the DNA polymerase. So this needs to, for adding the nucleotide, this needs 3' free hydroxyl group. So RNA primer, how do you get RNA primer? For the synthesis of a new DNA, short fragment of RNA primer, 5 to 50 monomers or nucleotides is required. So from DNA template, one RNA polymerase enzyme called as primase enzyme, it synthesizes an RNA primer. So it builds complementary base pairs to the DNA template. This enzyme doesn't need a 3' hydroxyl group. So it directly adds the nucleotides, which is complementary to the existing DNA template. So primase enzyme forms the RNA primer, and to this RNA primer, DNA polymerase starts synthesizing the new strand. So this is the RNA primer with 3' hydroxyl group. You can see here 3' hydroxyl group. Then one nucleotide is coming in with 3 phosphate group. This is the alpha phosphate group. These two are released as pyrophosphate. So this condenses with the free 3' hydroxyl group, forming the nucleotide backbone, forming the DNA, new strand. Then another nucleotide will come, the same reaction proceeds. This is how DNA polymerase adds on the nucleotides to the newly synthesizing DNA. The RNA primer is complementary and anti-parallel. We have already seen in the structure of DNA, the two strands are anti-parallel to each other. One strand is 5' to 3' and another strand is 3' to 5'. Now what is chromosome? Chromosome is primase and some pre-priming protein complexes together, they are called as chromosome. Now in this replication, you have on synthesizing of new strand by the DNA polymerase, two strands will be formed. One is the leading strand, which contains only one RNA primer, and another one is lagging strand, which contains multiple RNA primers. Now replication fork, what is replication fork? So replication fork consists of all these 4 components. That is unwinding enzyme, DNA helicase, then single strand binding proteins for preventing the re-annealing of the single-stranded DNAs, primase enzyme which helps in primer synthesis, DNA polymerase which helps in elongation of the DNA strand. So these are the 4 components present in the replication fork. This is the replication fork. Now you can see here, the DNA polymerase can add on nucleotides only to the 3' hydroxyl group. So the synthesis takes place only in the 5' to 3' direction. 5' to 3'. So the DNA template is read from 3' to 5' because both the strands are anti-parallel. So reading is from 3' to 5' direction. Synthesis is from 5' to 3'. Don't confuse this because DNA polymerase can add on nucleotides only to the 3' hydroxy group. So the synthesis takes place in the 5' to 3' direction. So now you can see in one of the strands where the DNA polymerase synthesizes in the 5' to 3' direction, it can synthesize continuously, no breakage. But in the opposite strand, you can see here, it can synthesize only in this direction, 5' to 3', because this strand is anti-parallel to this, which is 3' to 5'. So the DNA polymerase can read the template only in this direction and synthesize only in this direction. So new strand synthesis takes place from 5 prime to 3 prime. So what happens here? Once it unwinds here, so there is a primer and it will synthesize in this direction. And after finishing of this, the primase will synthesize a new primer here and it starts synthesizing and DNA polymerase elongates till it reaches the previous newly synthesized strand. Here it stops and then ligase will come and ligate these two. So these fragments are called as Okazaki fragments and this strand is lagging strand. I hope you have understood this. In mammalian genome, in one replication bubble, DNA synthesis occurs in both the directions. This is the multiple origin of replication cell. So in each replication site, there will be a replication bubble formation. Of this is the replication fork. So this is another replication fork. So here this replication fork extends like this, here this extends like this. So synthesis of DNA is continuous on the leading strand and discontinuous on the lagging strand. So short pieces of DNA are produced on the lagging strand. This is what is semi-discontinuous synthesis replication. So single strand binding protein, otherwise known as helix destabilizing proteins. They are not enzymes, bind only to the single-stranded DNA, keep the two strands separated, and also they protect the single-stranded DNA from degradation by nucleases. This is also very important because nucleases degrade the nucleotides. Now DNA polymerase, it has three important properties. One is chain elongation, that is the rate of polymerization. Then process safety. What do you mean by process safety? The expression of the number of nucleotides added to the nascent chain before the polymerase disengages from the template. That is, the DNA polymerase will not be disengaging from the template. So it holds on to the template and goes on synthesizing without disengaging itself from the DNA strand. And it has also proofreading capacity, that is, identifies the errors and corrects them. So DNA polymerase, synthesis of new DNA strand, synthesis occurs in the 5 prime to 3 prime direction. It's very important. Anti-parallel to the parent template strand, checks the incoming nucleotides, that is proofreading capacity. You can see here. So 3 prime end, it goes on adding nucleotides in this strand and synthesizing the new strand. The discontinuously formed small new DNA fragments are called as Okazaki fragments. These are then joined by DNA polymerase and ligase. Now you can see here the pole of replication. So this is the replication fork movement here. So in the 5 prime to 3 prime, when it is read along the 3 prime to 5 prime template, the strand becomes leading strand because it is continuously synthesizing. This is the topoisomerase relieving the supercoil. And this is the primase enzyme. This is the DNA helicase enzyme. And these are the single strand binding proteins. And now this is the RNA primer. So in the lagging strand, the RNA primer is here. So it starts synthesizing from 5 prime to 3 prime direction. Here you can see. So in this strand, 5 prime to 3 prime is from here to here. So it is continuously synthesizing. Here what happens, 5 prime to 3 prime is in this direction. So one RNA primer, it starts synthesizing. Now when it comes to this end, another RNA primer will be synthesized here. You can see here. So this is one fragment. And then another RNA primer is formed here, synthesized here. And then it is elongated by DNA polymerase till it reaches this end. So now after this, again DNA polymerase 1 and ligase. Because this RNA primer has to be removed. This RNA primer will be removed by DNA polymerase 1 in prokaryotes, and DNA polymerase alpha in eukaryotes. So this primer is removed and complementary base pairs are synthesized, and then finally the nick is resealed by DNA ligase. This is how replication occurs. So polymerase function here, proofreading capacity you can see here. So this is synthesizing 5' to 3' activity. You can see triphosphates, the bond is formed between alpha phosphate and 3' hydroxyl group. Bonding between, this is the complementary base pairs occurring, hydrogen bonding, pyrophosphate is released, and now, so this is the newly synthesized strand. Now proofreading capacity you can see here in this base pair, you can see instead of guanine, the nucleotide, incoming nucleotide that is bonded is adenine. For cytosine, complementary base pair is guanine. Here adenine is bonded. So what happens, because of the proofreading capacity means it has also 3' to 5' exonuclease activity. Why 3' to 5' exonuclease activity? Because sugar has to be removed from this end. Synthesis is 5' to 3'. So the wrong base has to be removed from this end. So the hydrolysis capacity is from 3' to 5' direction. So it has 3' to 5' exonuclease activity because it acts from the ends. Exonuclease activity. So because of this activity, this wrongly base paired base is removed. You can see here. And then correct base pair will be formed. So replication, I am repeatedly saying replication occurs in the 5' to 3' directions simultaneously on both the strands. So here you can see this is the primase enzyme. Primer is formed. So after finishing of this fragment, another primer will be synthesized here and it is elongated. Then helicase goes on unwinding. Then another primer is formed here and it elongates. But on the leading strand, what happens? It is continuously synthesizing the strand. So this is the leading strand and this is the lagging strand. So in this lagging strand, these fragments are termed as Okazaki fragments, and this primer is removed by DNA polymerase 1 and then nick sealing by DNA ligase. So replacement of RNA primer by DNA polymerase 1 in prokaryotes, RNA is H1 and flap endonuclease in eukaryotes, removes the RNA primer and then synthesized by DNA polymerase alpha. So it catalyzes the synthesis of small fragment of DNA in the 5' to 3' direction to be replaced in the place of RNA primer. And ligase form the phosphodiester bond. You can see here. So here RNA primer is removed and added. This is the phosphodiester bond formation. Here 3' hydroxyl group alpha phosphate. So you can see here. So this is resealed. This is done by ligase enzyme. Now in human replication, similar to prokaryotes, but thousands of origin. What is the difference? Thousands of origin of replication, multiple origin of replication, sufficiently fast, completed in 9 hours. Eukaryotic DNA polymerase less efficient because it is 10 times slower than the prokaryotic, since it has proofreading capacity also. And multiple replicating units are there. So this is in comparison of prokaryotic and eukaryotic DNA polymerase enzymes. So in E. coli, 3 DNA polymerase enzymes that are basically, so one is gap filling and synthesis of lagging strand that is done in eukaryote by alpha enzyme. You have 5 types. So alpha is for lagging strand gap filling, Delta is for processive leading strand synthesis. This is similar to DNA polymerase 3 in prokaryotes. Then epsilon DNA proofreading and repair, beta DNA repair, gamma is for mitochondrial DNA synthesis. So replication of eukaryotic, I've already told you. So this is in prokaryotes, origin of replication, single origin of replication, local opening, replication bubble formation. It is bidirectional, then daughter DNA is formed. This is in prokaryotic, very simple. In eukaryotic, multiple origin of replication, so formation of multiple replication bubbles here. Then finally, two new daughter DNA strands are formed. So after formation, what will happen? They have to be bound with histones for the formation of chromosomes. Because during replication, the chromosomes are relaxed and nucleosomes are loosened so that they can be replicated. So after replication, they have to be tightly bound with histones. So during separation, parental histones also associate with one of the parental strands. Then new histones will be with one of the newly synthesized strands. So one of the daughter chromosomes contain parental histones, the other one contain newly synthesized histones. So this DNA synthesis occurs in S phase, 8 to 10 hours. Methylation of DNA inhibits replication. So termination, how this replication is terminated. In prokaryotes, it has termination utilization substrate protein with termination sequences. They have TER sequences, that is termination sequences, which direct the termination after binding of this termination utilization substance. Because this prevents the helicases from unwinding of the DNA helix. Now in mammalian or eukaryotic DNA strand, what happens in the linear chromosomes, after replication, you have this 3' overhang at one end because, so here in the lagging strand, what happens? The final primer after removal, you have no DNA polymerase action at this end. So this will be shortened. After removal of this primer, no base pairs will be formed at this end. So if it happens in the successive division, what will happen? The chromosome, the DNA, chromosomal DNA will be shortened in the subsequent division of the cells. This results in aging. So chromosomes will be shortened. So to prevent this, we have the telomeres. So what are telomeres? Telomeres are complexes of non-coding DNA plus proteins which are located at the ends of the linear chromosomes. They maintain the structural integrity of the chromosome. They prevent attack by nucleases. So these are the properties of telomeres. They are short, repeat, non-coding hexameric sequence, GT-rich sequences. Telomeres have GT-rich sequences. So one another important enzyme is telomerase enzyme, which is a complex of protein which acts as a reverse transcriptase, because reverse transcriptase means they build or they synthesize DNA using RNA as a template. So short piece of RNA acts as a template here for reverse transcriptase enzyme. So G-rich strand is formed and this folds on itself to prevent the attack by nucleases. This is at the end of the DNA. You can see here, this is the eukaryotic DNA. After the removal of primer, here this end is shortened than this strand. Similarly, this end also. So this end, this is the telomeres. So you have telomere repeats, GT-rich sequences. This is the newly synthesized strand. Sorry. Newly synthesized strand with terminal RNA primer removed. Here you can see terminal RNA primer is removed. So then new telomere repeat has to be formed to synthesize strand, to elongate this strand till this end. So for this telomerase enzyme helps. This telomerase enzyme has one RNA template built-in RNA template. It has telomerase primase activity, so it synthesizes complementary strand to this. So CA-rich RNA template. So by the telomerase activity, it builds on synthesize this GT-rich sequences at this end, 3' end. So next what happens? RNA primer is synthesized by RNA primase at the opposite strand, and then it is elongated by the DNA polymerase. You can see here. So this is the RNA primer, which is built on by the primase enzyme. Then the DNA polymerase elongates this. So this is completed. Now you can have, this is the original strand length. So this is how eukaryotic DNA is prevented from shortening of DNA in the subsequent division, in the cell division. Now post-replication process, what happens after replication? Methylation of the new strand and proofreading, identification of the replication errors, correction of the DNA damage. These are the post-replication process. So prokaryotic and eukaryotic replication, these are the differences. I've already told you about the polymerase differences and circular chromosome and linear and foster processing activity. And here the replication is not controlled by cell cycle in prokaryotes, but during a phase of the cell cycle, replication takes place in eukaryotic. So cell cycle I'll take in my next class. So inhibitors of DNA replication. So three types. One is affecting DNA, which are intercalators and drugs damaging DNAs. Then affecting the substrates for replication. Substrates are DNTPs are one of the substrates. So inhibitors of production of this. Then substrate analogs, which lacks the 3' hydroxyl, they also inhibit replication, because for DNA replication, free 3' hydroxyl end should be there. Then affecting the enzymes which are involved in replication. So this is one assignment for you. You have to write the inhibitors of DNA replication. You should give example for each and everything. So these are some examples. Nucleoside analogs. So nucleoside analogs can be asked as short notes. Arabinose prevents the chain elongation. Cytosine arabinoside, anti-cancer chemotherapy, zidovudine and acyclovir. These are some of the examples of inhibitors. Nucleoside analogs, which helps in inhibition of DNA synthesis. So with this, DNA replication is completed. Thank you.